Assalamu alaikum, guys, again. We are going to start our next topic today. And what we are going to start today is about enzymes. That's the right timing to discuss enzymes because we will have a couple of practicals related to enzymes as well. So, by going through this topic, you will get an idea what enzymes are before you start the practical, just like you guys have already started the lipids and we have done two practical labs in relation to the lipids. So let me start the topic. How you define the enzymes actually? Enzymes are protein with specific catalytic properties in order to catalyze different metabolic reactions. And this is going on because of the power of their specific activation. So in short, enzymes are chemically protein and they have a specific activation capability by which they will catalyze the reactions, the metabolic pathways within the biological cell. In other words, enzymes are pretty much reciprocal to what the catalysts do in general chemistry. But catalysts are inorganic material. But we're talking about enzymes. Enzymes are chemically protein in nature, most of them. I'll discuss the exceptions later on. So we define the chemical reactions. We know that all the biological cells, from the prokaryotes to the eukaryotes and to the multicellular human being, they are full of chemical reactions. They are full of biochemical reactions, and there are thousands and thousands of reactions going on within a single cell. These all reactions are paramount towards the metabolic machinery of the cell. And in order to understand these chemical reactions, we must first understand what is activation energy. As it is written here that the chemical reactions, they require an initial source of energy, an initial input of the energy in order to carry out the reaction, in order to proceed with the reaction. And that particular input of the energy is termed as activation energy. So, activation energy is that minimum amount of energy which is required to give that input to start a metabolic reaction within the cell. Now, during this part of the reaction where we are giving this energy, the molecules are said to be in transition state. Transition state is the state which is in between the reactant and the final product. So, any metabolic reaction will definitely start with a substrate, which is a more appropriate term when it comes to the biological reaction, or in a general term, you call it a reactant in general chemistry. And that substrate or reactant will be transformed into a product. So, in between, there is a state called transition state. And the activation energy is actually the energy which is needed to convert a substrate into a transition state from where it will go on and transform into a final product. Now, this graph is going to give you a better understanding of what we are talking about. You must have gone through this graph earlier in your chemistry courses. So you can see here that this is what the reactants are, what we call the substrate, and that's the product. Now, in this particular graph, you can see that this reactant or substrate has got some amount of energy which is already been there in the reactant. So that energy, which is called the free energy, is the energy which the reactant molecules already have it. Now, in order to be converted to react from the reactant to the product, you can see that there is a particular amount of energy needed. This particular amount of energy needed to transform those reactants, which already have some energy, to be transformed at the transition state. This particular energy I refer to activation energy. And from the graph itself, it's very clear that once the reactant are at this energy level, then they are going down. So that means they don't need any energy. Rather, from the transition state to the final product, there will be the release of energy as you look at the graph. So the reaction pathway has got this particular graphic representation where a reactant would have some sort of energy already within the molecules. That's called the free energy. On top of that, the energy which is needed to bring those reactant molecules up to the transition state is called activation energy. And from the transition state, you can see as it's going down, that means it's spontaneous and that is going to be transformed into a product. In other words, I can say that if the reactant is being transformed to transition state, then it must go down to form the product. Otherwise, if it is just kept here without that particular input of the energy, we cannot expect that reactant to be transformed into a product. So this is a particular reaction pathway. Another important aspect of these enzymes and their role is to make the reactions go faster. So there are two different things to be kept in mind. One, you need specific amount of energy to carry out a reaction, which is a must condition. If you don't provide that energy, the reaction will not be carried on. And the other thing is that the reaction speed is going to be fast. It's increased. So these are the two different aspects that you have to consider. Now, there could be other factors, there could be other changes that you can do in order to increase the reaction speed or in order to, enhance the reaction speed. So, one thing is increasing the temperature. So, if you can increase the temperature, what it results? It results in the increase of the kinetic energy of the molecule. Or in other words, I would say that increasing the temperature will increase the free energy, as you have seen in the graph previously. This one, that's the energy already possessed by the reactant. So when you heat it, when you increase the temperature, the kinetic energy of the molecules increase. In other words, the free energy of the reactant molecule increases. And that's how it moves fast. So in biological systems, they are very sensitive to temperature changes. Like in general chemistry, you can increase the reaction speed by increasing the temperature in a controlled way. But that's not going to be a choice for us when it comes to the biological system, because we all know that within the cell, within the biological system, the temperature are pretty much constant, like that, what we call the normal body temperature. So we do not have much of the choice to increase the temperature to enhance the speed when it comes to the biological reaction. But we can do that in general chemistry when we are doing some reactions in the lab or outside the body. Now, enzymes are the one that can overcome this thing. Enzymes can increase the rate of reaction without increasing the temperature. So here comes the significance of the enzymes in the biological system, that they not only provide the activation energy, they also increase the rate of the reaction, for which you don't need increase in temperature. As I requested earlier, and I'm going to repeat this last time, that people please join the class on time. You must join the class on time, what the time has been given. Otherwise, it's going to disturb our class, and I'm not going to go back and add you people in the class. So this is the last warning. Next time, I'm going to strictly follow the time, and after which, if you enter the class, I'm not going to enter you because it's disturbing the class in between, because I have to switch the screen in order to add you in the class. So please make sure next time there should be no delay when it comes to the schedule time. I hope you understand. So the enzymes, what they do, like how they increase the rate of the reaction, the process or the mechanism by which the enzymes increase the rate of the reaction without changing the temperature is by lowering the activation energy. So what I can say in this picture if this is the activation energy required under the normal circumstances. In presence of an enzyme, for example, it may decrease up to this level, half. So that means a little amount of energy is needed to transform the reactant into transition state. And that's how the reaction speed would be faster. So the one way how enzymes can accelerate without changing the temperature is that enzymes actually lower the activation energy requirement for that particular reactant to be transformed into the transition state. So that means they create a new reaction pathway, which will be referred to as a shortcut. Now look at this picture, that gives you an idea. Under the normal condition, this was the activation energy required to be transformed into final product. But in presence of the enzyme, this dotted line indicates an alternative pathway where you can see the activation energy has been reduced. So that's how the enzyme model works, that it reduces the requirement of the energy of the activation, and thereby it enhances the speed of the reaction. So enzyme control reactions, they normally proceed 108 to 1011 times faster than corresponding to a non-enzymatic reaction. So, as I said, that in biological reaction, we cannot achieve this faster speed by just changing the temperature, because that's not possible in the biological system. But enzymes can do that, and they can enhance the speed of the reaction by many folds. As said earlier, that enzymes are chemically protein, and you already have studied about the structure of the protein in 311. You know that proteins, they have primary, secondary, tertiary, and quaternary structure. You know the levels of these structure play a vital role towards the functionality of the protein. A primary structure of a protein and a secondary structure of the protein is not a functionally active protein. For a protein to be functionally active, it must be either tertiary or quaternary level. So enzymes are protein, they are functional protein. They have the role to catalyze the metabolic pathways, and they have a specific 3D structure that corresponds to their tertiary or their quaternary structure. Now, the picture in front of you is of a very common enzyme, which is human pancreatic amylase. And I'm sure that you know that this amylase is the enzyme that is involved in carbohydrate metabolism or carbohydrate breakdown, and that's specifically of pancreatic region. So you can see that it's a 3D structure being shown for the enzyme. Next thing, when we understand that the enzymes are proteins, we know that the proteins have a particular structure, as I said, and that involves a particular folding as well. Like if we start from the protein, we have a linear polypeptide chain, which is a primary structure where we have only the peptide bonds. Then we have the secondary structure where we have hydrogen bonding and a couple of other forces involved. Then we have the tertiary structure where we have the disulfide linkages and many other hydrophobic interactions. And then we have the quaternary structure where we have got multiple chains. So we all know that for a protein to be functionally active, there is a specific formation, there is a specific, I would say, folding, which is a must condition for a protein to be functionally active. Now, no matter it's an enzyme or any other protein, any kind of protein to be functionally active must undergo a proper conformation, a proper 3D structure. So when you look at the enzymes, they are protein, and definitely they do have a proper 3D structure. In that 3D structure, they have specific sites called the active sites. These active sites are of particular importance. The shape and the chemical environment inside the active site is actually going to allow a chemical reaction to proceed more easily. So what I want to say is that in case of an enzyme, the active site is that particular site which actually bind with the substrate, which actually bind with the reactant. And the binding of the reactant with that active site is actually going to facilitate the reaction. Let me clarify it further. Like if you look at the enzyme, it's a big molecule because it's protein. But that big molecule has got a specific site which is called active site, and if the reactant is going to bind with that active site, thereby the enzyme can catalyze the reaction. So this is going to be a model where you can feel like, the enzyme is a very big molecule in contrast to the reactant or the substrate molecule, which is very, very small as compared to the size of the enzyme itself. The next important thing is to discuss about cofactors. If you remember, we had a discussion about the cofactors when we discussed about the minerals and vitamins. And I told you that there are many minerals specifically which are required in our body to be, function as cofactors. What they do, like for example, you have an enzyme, you have a particular substrate, the substrate is going to bind with the active site and thereby the reaction is catalyzed. But this reaction cannot be carried out until unless there are some other essential requirements. And one of the essential requirements for many of the enzyme catalyzed reactions are the cofactors. So cofactors are kind of essential requirement in absence of which the enzyme is not capable of catalyzing the particular reaction. So the cofactors are an additional non-protein molecule, which, as I said, most of the cases are minerals, that is needed by some enzymes, not all, to help the reaction. What these cofactors are doing, they're tightly bound with a specific enzyme with a prosthetic group, a specific group. The cofactors that are bound and released easily are called coenzymes. So cofactors have got another category called the coenzymes, where they can bind with the enzyme and they can be released from the enzyme. So it's a kind of a reversible process where they bound with the enzyme when the enzyme has to catalyze reaction, and once the reaction is catalyzed, they gonna dissociate from the enzyme. So we call them coenzymes. As I said, many of the vitamins are the coenzymes. As you can see in the picture, nitrogenase is an enzyme, and the cofactors for that enzyme are iron, molybdenum, and ADP, adenosine diphosphate. The next term is substrate, which I already has discussed, and the substrate are actually the reactants in general chemistry term. Substrate of an enzyme are the reactants that are activated by enzymes, or in other words, going to be transformed into product. So enzymes are specific to their substrate. Now that's a very, very important point, and that has to be kept in mind. That uh, we have a bit of specificity when it comes to the enzyme catalyzed reaction. A particular enzyme is very much specific for a particular reaction. In other words, a particular specific enzyme is specific for its substrate to catalyze the reaction. And this specificity of the enzyme is actually determined by the active site, which I mentioned earlier. So that means a particular enzyme has got a particular active site that is capable of binding with a specific substrate. So not all substrate can bind to the active site, and that's how enzymes are highly, highly specific. Okay, till now we have gone through some basic concept about what enzymes are, cofactors, substrate, reactant, activation energy, and so on and so forth. Now we are heading to the next step of the discussion, which is another very important part of the discussion in enzyme catalysis, and that's called the lock and key hypothesis. Now, this lock and key hypothesis is a hypothesis which is being proposed to explain the enzyme catalyzed reactions. And I think the name itself is pretty much self explanatory, as you can see from this, the lock and key model. So what actually happens, the substrate is going to fit between the active site of the enzyme. So that means the enzyme active site and the substrate, they are like lock and key. They are like, made in a way that they are, how would I say, like they are compatible. Just like A lock and a key are compatible. In a similar way, the active site of the enzyme and the substrate, they are compatible. That's how we call it a lock and key model. So the key is analogous to enzyme, and the substrate is analogous to a lock. So the temporary structure of the enzyme called enzyme substrate complex is formed. So once the substrate is bound with the enzyme via the active site, this is called enzyme substrate complex. And why we call it temporary? Because it has to be transformed into final product. So this is a intermediate kind of a stage where the enzyme is bound with the substrate, and we call it enzyme substrate complex. Products have a different shape from the substrate. So once the substrate is transformed into a product, that means it's no longer compatible with the active site based on the lock and key hypothesis. And that's why once the product is formed, it quickly dissociates from the enzyme because it's not compatible. So I think that's pretty much easier to understand. So that's how once the product is formed, it's released from the active site. Once it is released from the active sites, that means the enzyme is again available to catalyze another reaction. So this is the picture explaining the whole process. You can see this is enzyme and this is substrate. This is called the lock and key model. They are complementary. They bound. This structure is called enzyme substrate complex, which I said is a temporary formation. And from there, the reaction is catalyzed and you got the product. Now the product shape is different from the substrate. That's why they get dissociated. And now you got the free enzyme again, which can be utilized for further reaction. The lock and key hypothesis explains how the enzymes are specific, as I said earlier. And this explains the loss of activity when enzyme denature. Because as I said earlier, that enzymes are protein in nature. And you know the protein functions, that if for any reason, uh, if the protein undergo any uh, unfolding, or protein will undergo any change in the structure because of any change in the conditions, we call that this protein becomes denatured. Specifically, like when you heat the protein up to a high temperature, the structure is distorted, and we call it a denatured protein. So once the enzymes are also protein, when they got denatured, definitely their activity of binding with the substrate is also lost, and that's how they are no more capable of catalyzing the reactions. There is another model beside the lock and key model to explain the enzyme catalysis, and that's called the induced fit hypothesis. The name itself again indicating induced fit. That means something will come and bind, and as a result of this will brought about the change. It unlike the lock and key model which you studied. So in some proteins, they can change their shape or they can change their conformation. So when a substrate is going to combine with an enzyme, it brings about a change in the enzyme's conformation. And as a result of that, the active site is then molded into a precise conformation. So that means the binding of the substrate will result in a conformational change of the enzyme structure, which in turn make a conformational change to the active site, making the chemical environment suitable for the reaction. Now, the bonds of the substrate are stretched to make the reaction easier. That means by lowering the activation energy. So I think the concept in both the lock and key model or the induced fit hypothesis is same in terms of the fact that it lowers the activation energy. But the difference is that in the induced fit hypothesis, it says that the binding of the substrate will trigger the conformational change in the enzyme, which in turn will mold a more precise conformation of the active site, thereby making the environment suitable for the reaction to be carried out. So this is called the induced fit hypothesis, and this is the picture where you can say that hexokinase A and B with or without the substrate, which is glucose molecule. So as you can see, this one and this one. So binding of the substrate results in the conformational change, while without that, it is an open site. So this explains the enzymes that can react with the range of the substrate of the similar type. Now, in the lock and key model, the limitation would be specificity because it's not changing. While in this case of the induced fit hypothesis, one can say that the process can explain that enzymes can react with a range of the substrate because it's not a lock and key. Some other substrate like similar to that may also come in and catalyze the reaction. And this is somehow can be explained by, for example, there are certain, like this enzyme. This enzyme is hexokinase. Now the term hexo refers to six. There is a specific enzyme called the glucokinase, and the glucokinase, as the name itself indicating, is specific for glucose. There is a fructokinase as well. But when we talk about the hexokinase, we can see from the name that it's not a specific enzyme. It's a general enzyme. Hexokinase can catalyze the reaction of the glucose, fructose, or maybe some other six carbon sugars. So the induced fit hypothesis actually correlates with this kind of reaction where more than one substrate can bind with one enzyme. Now, coming to the next point, that what are the factors affecting enzymes? We studied at what enzymes do. We studied about the model by which they can bind with the substrate. We know that enzymes catalyzes the reaction by lowering the energy of activation, and that's what we all studied. Now, the next step is about the factors which can affect the enzyme activity. So there are four factors we are going to discuss here: substrate concentration, pH, temperature, and inhibitors. Let me go through them one by one. And this is an important topic because, as I mentioned in the beginning, that these sort of factors affecting the enzymes is also the part of our labs. So we'll be doing some labs in near future where we see how enzyme activity is affected by substrate concentration, pH, temperature, and inhibitors. So first is the substrate concentration. Now look at the graph. We have on the y-axis reaction speed, and on the x-axis we have substrate concentration. This typical graph that is in front of you is called a straight line graph, and this graph is an indication of direct proportionality. So this is a graph of substrate concentration with non-enzymatic reaction. So we see that the increase in the velocity is proportional to the substrate concentration. So it's a very simple straight line graph which called the direct proportional. More, if you increase the substrate concentration, then it will increase the reaction velocity. So this is pretty much, I think, simple. There is nothing to explain more in this graph. Now, this is the same graph of a substrate concentration, but in presence of an enzyme. Now, you can compare this graph with this graph. Now, in this case, it's a simple straight line. More substrate, more reaction velocity. This kind of the graph that is in front of you, you might have seen that in general chemistry lab. When you are doing the reactions, in chemistry lab, normally it's being seen that if you increase the reactant concentration, then there will be more product formation and more reaction speed. This is the general law of thermodynamics when it comes to an ordinary chemical reaction we are doing in the lab. But when it comes to the reaction in presence of the enzymes, there is a little difference. The difference is, like you can see, with the increase of the substrate concentration, the reaction velocity is also increasing. Now there are two things to figure out. One thing, the speed of the reaction increasing in this case is lesser, while the increase of the reaction velocity with this in presence of enzyme is much faster. You can see it's going like this instead of without enzyme where it was going like this. So increase of the reaction speed has been observed in both, but here the proportion of that is higher as compared to without enzyme. Second important point is this graph goes straight. That means if you keep adding substrate, the reaction velocity is going to increase and increase. But here, after a particular point, which is called the Vmax, the maximum velocity, you see that there is a straight line coming. This straight line is indicating that there is no more increase in the velocity, no matter you increase more substrate. So that means when you add the substrate, the reaction velocity increases, but when it reaches this point, then adding more substrate is not affecting the velocity. So faster reactions, but it reaches the saturation point, as I said. When all the enzyme molecules are occupied, there will be no change, no matter you add the substrate. So, this is how we explain as compared to the mechanism I shared with you, that substrate is going to bind with the active site. So for example, if you've got one big enzyme molecule, let's say, and it has got two, three, four, five active sites, and you added the substrate, so they are going to go and bind with the active site. Once the enzyme is fully saturated, that means no more active site is available. So once it is saturated, if you add more substrate, it's not going to increase the reaction velocity because enzyme is saturated. So that's how the model is explained. If you alter the concentration of the enzyme, then the situation is different, because then the Vmax will be changed also, because adding more enzymes means more active sites and more available spaces for the substrate. So that's a different story. The second is the effect of the pH condition. Now, effect of pH condition is also another very important factor that's going to affect the speed of enzyme catalyzed reaction. Now, if you look at this picture again, on the x-axis we have enzyme activity, and on the on the y-axis we have enzyme activity, and on the x-axis we have pH. By the way, you must know that it's an arbitrary thing. You can call this as x-axis or this as y as well, but just in most cases we call this y-axis and this x-axis. So here we have the enzyme activity, and here we have the pH, and we have the example of two enzymes here, trypsin and pepsin. Pepsin is an enzyme which is involved in protein catalytic reactions, while trypsin is another enzyme which is involved in the protein catalytic reaction. But their origins are different. The pepsin is an enzyme which is present in our stomach, and that's a very highly acidic environment because of HCl, while the trypsin is in the mouth or in the gut. So in the mouth, the situation is more alkaline. In the stomach, the situation is more acidic. Now, this graph clearly indicates that the effect of the pH on the enzyme activity primarily depends on the enzyme itself. That means some of the enzymes would have their good activity or maximum enzyme activity in the acidic pH, and some of the enzyme would have their best activity in the alkaline pH. So that means enzyme itself has got this nature that whether they are active in acidic pH or alkaline pH. So there is no one thumb rule that you can apply, just like in the substrate, that increasing the pH will increase the activity or vice versa. We have to look at the enzyme itself. So in case of the pepsin, its maximum activity is being shown in pH between 1 to 2. And as you increase the pH, what's going on? The enzyme activity is decreasing, ultimately reaches the point where there is no enzyme activity. So that means pepsin is not going to work under the alkaline conditions. While in contrast, the trypsin has the minimum activity at enzyme pH 3, which is acidic. And as you increase the pH, it reaches the level of about between 7 to 9 pH. It has the maximum activity. And then increasing further pH is not going to increase the activity, it's going to decrease. So this particular graph is very much explanatory that effect of the pH will be varied upon depending upon what enzyme we are talking about. So in case of the effect of the pH, the extreme pH level will produce the denaturation. Now, extreme pH level means both extreme acidic and extreme alkaline. As a result of this, the enzyme, because it's a protein, it undergoes denaturation. Denaturation is the breakdown of its hydrogen bond. As a result of the denaturation, the conformation of the protein is changed. And we all know that when the conformation of the protein is denatured, it's no more capable of catalyzing the reaction. The structure of the enzyme is changed because of the extreme pH. The active site is also distorted and substrate molecule will no longer fit in it. At pH values slightly different from the enzyme's optimum, even though enzyme is capable of catalyzing the reaction, but there is a small change in the charges of the enzyme, and that will result in the decrease or increase of the rate. This change in ionization will affect the binding of the substrate to the active site as well. So there is one thing called the optimum pH. If you look at this graph, we have that optimum pH of pepsin is 2, and the optimum pH of trypsin is 8. So, but we can see that if it's pH 7 or pH 6, it is working, but that's called the optimum pH where we have got the maximum activity. So if you keep the pH either on this side or on this side slightly different from the optimum, even enzyme is capable of catalyzing the reaction, but there is a change in the ionization because of the pH change, and the change of ionization would affect some sort of changes in the binding of the substrate with the enzyme, and thereby affecting the speed of the reaction. I'm going to stop here at the effect of the pH, and I'll discuss the rest of the two effects in my next class. Meanwhile, I'll share the slide with you, and you can, go through it and ask the question later on. Or if you have question at this stage, you can raise your hand and then ask the question. I'm here to give you the answer. So the forum is open for the question. If you have any question, just raise your hand. Okay, so if you don't have any question, so I'm going to finish the lecture here, and inshallah we'll share the recording with you guys once it is available. Thank you very much, and Khuda Hafiz.